Hey, welcome everyone. I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I would also like to pay my respects to elders past, present, and emerging. Well, good afternoon and welcome to the April FightMND Scientific Seminar. My name is Sarah Bennett and I'm the Research Coordinator at FightMND. We hold these seminars monthly on the last Tuesday of the month, and last month in March, we brought you the team from the Australian Research Data Commons. They highlighted some of the ways in which this national research data infrastructure can support M&D research. If you would like to know more about the work of the ARDC and how it might support your projects, you can watch a recording on our website now. Following the talk today, we are leaving plenty of time for discussion and questions. So if you would like to ask anything, please use the reaction button to raise your hand and one of our team will enable your microphone and camera and let you know to ask your question. Alternatively, you can also type your question into the chat where our team will be monitoring and can read out questions on your behalf. Please note that the chat function is only enabled if you're using the app-based version of Teams and unfortunately doesn't work in the web-based version. Please ensure you do introduce yourself and where you're from before asking your question so we can all get to know each other. So now to our presentation for today. Associate Professor Cheyenne Ngo is a group leader at the Australian Institute for Bioengineering and Nanotechnology at the University of Queensland, Director of the UQ Centre for M&D Research and an affiliate adjunct academic at the Royal Brisbane and Women's Hospital and Murdoch University. Associate Professor Ngo transitioned into M&D research after receiving her PhD in neuroscience from the University of Queensland in 2009 and started her independent research group in 2015. She's been awarded numerous fellowships to support her research program, including being one of the inaugural recipients of a FightMND Mid-Career Fellowship in 2020. Associate Professor Ngo leads a translational research program that strongly incorporates people with lived experience of MND, and she's here today to talk through her journey of bringing a bench discovery into clinical trial. Thank you so much, Sue, for taking the time to present to us today, and over to you.
Thank you, Sarah, for the introduction. You can see my screen okay?
Yes, all good, thanks.
Yeah, okay, good. Thank you. So I too would like to acknowledge the traditional owners of the lands on which we meet today and pay my respects to elders past, present and emerging, as well as those who did not make it to elder status. And thank you for joining me online today. What I'd like to sort of take you through is my journey from discovery research through to clinical trial in the context of ALS. And all of this would not really have been possible without support from the M&D community and M&D associations, including FightMND and M&D Australia. But before I start, I would like to acknowledge people in the team. It's only really the people in the A team who, work hard day in, day out for the common goal of a world without ALS M&D that we've been able to get to where we have been. So a lot of the work that I'm presenting today really is come off the back of the hard work of the team, past and present members. Now, in terms of motor neuron disease or ALS, amyotrophic lateral sclerosis, I'm sure a lot of you would have heard that, it's a highly heterogeneous disease, and it's really characterized by the death of the nerve cells that control our voluntary movement that are located up in the motor cortex and in the anterior horn of the spinal cord. Now, the heterogeneity of the disease is really exemplified through differences in not only site of onset or progression between people who are diagnosed with this disease, but also survival, with survival on average being anywhere between 2 to 5 years, although some people may pass away with M&D much earlier and some much later. There are a lot of things that are proposed to really drive the heterogeneity of M&D, and some of these really probably are associated with the pathogenic mechanisms, or those things that are thought to contribute to the onset and progression of disease. And this can include abnormal protein aggregation or inflammation. One of the main things that we focus on in the lab at UQ is differences in metabolism. And what we know in M&D is that the capacity for somebody to tolerate the metabolic impacts of the disease can really dictate where they fall in terms of their progression and their survival. We know that decreases in body weight in people with MND ALS is linked to worse prognosis and outcomes. A lower BMI is also linked to poor prognosis. Loss of fat mass throughout the course of disease is linked to shorter survival, whereas people who have more fat mass may have longer survival. And so, this background of this metabolic dysregulation and metabolic differences in people with MND is really what led us to move into an area of research that eventuated in what I will present today, which is the METFLEX clinical trial. But in order to run or get to that point of a clinical trial, there were a lot of things that we had to do in the lab in terms of our research to be able to put pieces of the puzzle together to do true translational research. So in 2015, with Derek, I established a natural history study working very closely with people with MND and ALS, where they would come to the clinic every 3 months and contribute to research, where we would look at how their body uses and generates energy. And over the years, we have transitioned from using just mouse models of the disease through to using stem cell-derived cultures. We grow motor neurons in a dish, through to organoid models of the disease that are also made from the same stem cells, to cells that are collected from the human blood samples and postmortem tissue. And it's the connecting of all of the data that we generate across these models and from people with MND that has allowed us to pursue certain research directions that have set up the clinical trial. Now, in terms of altered metabolic balance in, let me see, in MND, I think the first evidence that there were changes in energy balance really came in 2004, which was this landmark study by Luc Dupuy and his supervisor at the time, Jean-Philippe Lafleur. And using a mouse model of MND, they showed a few key things. The first being that body weight in the SOD1G86R mouse model of MND was lower than mice or wild-type mice that did not have the disease. And they also showed that body weight was also lower in the SOD1G93A mouse model of the disease. And they also showed that energy expenditure in the G86R mouse was much higher, and this is represented, the ALS mouse here is represented by the black bars. Pre-symptomatically, before these mice developed ALS or MND-like phenotypes, and when the mice then did have ALS phenotypes. And they also showed that this energy expenditure was also higher in the SOD1G93A mouse. This was the same story for both total energy expenditure as well as resting energy expenditure. And this is where this concept of hypermetabolism, of increased metabolic rate, was first really addressed in the preclinical setting. And then a few years later, in 2000, well, actually 6 years later, I invited myself to Jean-Philippe Lafleur's lab just to do some research related to metabolism in MND. And we came up with this idea that perhaps metabolic changes in the disease was something that we could target. And we initiated some research back in 2010, which later accumulated and eventuated in this publication in 2015, where we showed that a metabolic switch in the use of glycolytic substrates in glycolytic muscle was preference towards lipids. So what this really means is that in skeletal muscle that is highly preferring sugar as the energy substrate, for some reason in these mouse models of ALS, the muscles were turning away from sugar and wanting to use fat as a fuel source rather than using the sugar. And the way we showed that this was the case was we ran SOD1G86R through an exercise paradigm where they either did intense exercise or endurance exercise. And under intense exercise, the SOD1G86R mice, or the ALS mice, performed worse than the non-ALS wild-type mice. Whereas if we put them through an endurance exercise paradigm, sort of like running a marathon as opposed to running a 100-meter sprint, they actually performed better than the wild-type mice. And this is important because the difference in the fuel substrates that muscle uses when doing intense exercise versus endurance exercise is very different. For intense exercise, the muscle will generally use sugar, whereas for endurance exercise, the muscle will turn towards using fatty acids or lipids because they are a higher source of energy a higher fuel source for energy production. And so this was the switch that we saw occur in these mice. Now, in normal instances, as I've said before, muscles will generally use glucose as the energy substrate or the sugar, and this leads to energy production in the mitochondria. And when glucose is not available, when sugar is not available, cells will turn towards fat to use as an energy substrate, and they can generate energy in the mitochondria that way. But what we found in the ALS mice, or MND mice, is that there was this preferential use of the fatty acids. And while fatty acids or lipids are a really good fuel source, one problem with using fat as a fuel source is that over time, it takes more oxygen to break down each fat molecule to generate energy, which means that you will end up generating more reactive oxygen species in the cells, and this eventually becomes toxic. Now, knowing that this switch was happening in these ALS mice, we'd come up with an idea of maybe it was something that we could target. And in this 2015 manuscript, we showed that a drug called dichloroacetate, which has been used in the cancer setting and which actually promotes cells back towards using sugar as opposed to lipids, we showed that that drug was able to improve function in these mice. But in this study, we actually fell short of measuring survival just because of the time course of the study. Now, if we can understand that a drug that changes the way that cells can access energy might be improving function in these mice, the next question really is this something that we can do in the clinic? Can we translate this information that we've found in preclinical mice into something that is clinically meaningful for people who are living with MND? So what we know in MND now is that even in people living with this disease, as well as the mouse models, is that there is this concept of an increase in resting energy expenditure. Now, you may have learnt back in primary school that energy balance equals energy in plus energy out plus growth. But what we think is happening in MND is that there may be a decrease in energy intake, whether that be through the diet or other means. But there is this increase in energy expenditure or energy use. And this leads to a compromise in energy balance, leading to weight loss. Now, in terms of energy expenditure, there are three main components that make up total energy expenditure in a living human. 10% is due to dietary-induced thermogenesis, or the energy that is expended through the burning of your food. 20% is associated with activity. So this is called activity-dependent energy expenditure. And then the remaining 70% is what your body would use at rest. Now, I had mentioned before that that study in mice in 2004 was the first real indication of this hypermetabolism in ALS, at least in the preclinical setting. But actually, the concept of hypermetabolism in people living with MND was not new. It was something that was known back as early as 1996. In fact, pioneering research done in 1991 by a Japanese group showed that people with ALS who were on mechanical ventilation were hypometabolic. So they actually had lower metabolism. But since that time, from 1996 up until 2009, several groups in the US and in France showed that people with MND had this higher metabolism. And sometimes this metabolism was seen in about 50% of people with sporadic disease, but in 100% of people who were studied who had familial forms of MND. And even though there were many studies that showed that hypermetabolism was a feature in people living with this disease, the clinical significance of hypermetabolism had not really been established even up in 2009. And so in 2015, Derek and I initiated a study, a natural history study, to follow people with MND throughout the course of their disease to see whether or not, one, they were hypermetabolic, and two, how hypermetabolism might actually be related to clinical outcomes. And this study also involves recruitment of healthy non-ALS individuals so that we can compare hypermetabolism in MND ALS versus healthy controls. So this is a bod pod, which is used to measure body composition. And from here, we can predict how much energy someone should be using. And then when people lie down on the bed and have this canopy over their head, we are measuring carbon dioxide oxygen exchange. And through the ratio of oxygen consumed relative to carbon dioxide produced, we get a measure of how much energy someone is actually using. And we published the first cohort study in 2018. And one, we did show that people with ALS MND were more likely to have this high metabolic rate compared to people with without the disease. But there were two key pieces of data that we were able to generate in this study. The first was that those people with MND who had hypermetabolism, or this increase in metabolic rate, which is represented in these graphs by the pink line, they had a faster disease progression. So the decline in their ALS-FRS-R functional rating scale was a lot faster than those people with MND who did not have hypermetabolism. And I think the key piece of information that we generated from this data was that we found that those individuals who had hypermetabolism had a doubled risk of death in the first year of coming to see us for a metabolic visit. And so people with hypermetabolism had worse survival outcomes compared to people with MND ALS who did not have hypermetabolism. Now, since our study in 2018, many more people have gone on to show that hypermetabolism is a feature in cohorts of ALS patients across the world, including in Japan and China. And recently, there have been studies, even in 2023, there have been 2 studies looking at hypermetabolism in people with MND. So now that we know that hypermetabolism is a feature of disease in people with MND ALS, there are a few questions that we wanted to ask. One is, what might be the cause for hypermetabolism? Because if we know the cause, then potentially we can identify the target for treatment. Now, in our 2018 study, what we did show was that people who were hypermetabolic had an increased lower motor neuron score. And this is a scoring system that some neurologists use in the clinic to really define the involvement of the motor neurons in the spinal cord. So a score of 0 to 2 means that there's minimal involvement. 3 to 6 means that there's more involvement of the motor neurons in the spinal cord, whereas 7 to 12 means that there's significant involvement. And by involvement, it's not something that you can measure by, cutting open the spinal cord and having a look. It's mainly looking at the clinical features that someone presents with and grading that on a scale of 1 to 3. And so the addition of those scores over the 4 limbs, the upper arms, the arms and the legs, will give you a score of 12. And so what we noticed is that people with MND who had hypermetabolism had quite a high lower motor neuron score. Now, given that the lower motor neurons in the spinal cord are the ones that project out and send connections to skeletal muscle, and knowing that we'd shown in the mice that skeletal muscle seems to have this switch towards using a fuel substrate that requires more oxygen to burn, we figured that perhaps hypermetabolism was originating from skeletal muscle in MND ALS. And so to really answer this question, we had to take this knowledge that we learned from people living with this disease back into the lab. And to answer this question, we used mouse models of the disease, but we also used muscles that we were able to generate from donations of people who were involved in our studies. So people with MND-ALS who were part of our studies would donate a skin and muscle sample. From the skin sample, we could make stem cells, which we could turn into neurons so that we could use that to study energy use in neurons. But from the muscle, we were able to grow up the muscle stem cells, turn them into muscle fibers in a dish, and study how they were using energy substrates, that being sugar and fat. Now, in terms of our mice, we had to establish capacity to be able to measure energy expenditure in mice. And so we developed, we wrote a grant which was funded and was able to support the purchase of some of these big chambers. They're called the phenomasters, but they're essentially little houses or cages that mice can sit in, and you can measure how much energy they're using, how much food they're consuming, how much water they're drinking, and how much they're moving. We also got a seahorse, which is a very fancy and very expensive machine that can measure how much energy cells are using in real time. And by combining these two pieces of equipment, we were able to study the mouse tissue, but as well as cells that we'd derived from people living with MND, as well as healthy controls. Now, in terms of the SOD1 mouse, we used the SOD1 G93A mouse model of the disease, keeping in mind that in 2004, Luke had already published data to suggest that the SOD1 mouse had hypermetabolism. We went to replicate this work. So we've got 3 graphs here. PS is for pre-symptomatic, OS is for onset, and MS is for mid-stage. And this really ages that define predefined stages of disease in the SOD1 mouse model, where it has no disease phenotypes, the onset of the disease phenotype, or more severe disease phenotypes. In these graphs, the blue line or the teal line represents the SOD1 ALS mouse, whereas the black line is the non-ALS wild-type mouse. These traces are of oxygen consumption over a 24-hour period. The white portion of the graph is the light cycle, and the dark portion of the graph is the night cycle. Because mice are nocturnal, you would expect that their energy consumption is higher at night because that's when they're more active. And what we can see is that throughout the progression of the disease, oxygen consumption or energy expenditure is increased in the SOD1 G93A mice by the time they get to the mid-stage, where they have quite a significant MND phenotype. But at the onset and the pre-symptomatic stages of the disease, they're very similar to the wild-type controls. And if we quantify this, we can see that, again, pre-symptomatic and onset, the SOD mice and the wild-type mice are essentially very similar, whereas by the mid-stage of the disease, the SOD1 mice have high oxygen consumption, and therefore we, by proxy, assume energy use over a 24-hour period, which is made up by increased oxygen use over the light cycle and the dark cycle in a 24-hour period. So now that we can confirm that the mice are hypermetabolic, we wanted to know whether it was the muscle that was causing this problem. And so we isolated muscle from pre-symptomatic onset and mid-stage mice. The SOD again here is in the teal and the wild type is in the white. And we can show that the dependence of these muscles on using sugar as the energy substrate is very similar in wild type and ALS mice. But when we look at how dependent the muscle is on using fat as an energy substrate, we can see that while at pre-symptomatic and onset of the disease, the SOD, ALS mice and wild-type mice are very similar, by the mid-stage of disease, you have this much higher dependence on the use of the fat as the energy source in the ALS mice compared to the wild types. And this is all good and well in mice, but is this something that we see in muscle from people living with the disease? What we did was we took those muscle stem cells from donors with ALS, but also healthy controls, and we grew those into muscle fibers, and we looked at how they used sugar and fat. And when you compare the ALS samples to the control samples, you can see that their ability or dependence on using sugar as the energy substrate is very similar. Whereas when you look at the ALS muscle and how dependent they are on using fat as the energy substrate, you can see that it's much higher than that in controls. And so similar to what we've seen in mice, we're seeing a very similar phenomenon in people with ALS, but also in the muscle from people with ALS. And so when we think about targeting this metabolic flexibility or this balancing of the use of sugar and fat as energy substrates in muscle, there are a few things that we did. So in 2013 was when we used dichloroacetate, and we published that paper in 2015 showing that dichloroacetate improves function, but we weren't able to look at survival. We then started pursuing other drugs that are approved for use in other conditions that might help tweak the way that cells access energy. In 2017, we came across ranolazine. Ranolazine is approved by the FDA, TGA, and EMA to treat angina. And what ranolazine does is it works here in the mitochondria where it completely shuts down the cell's capacity to access fat as the energy source. And we showed that ranolazine, again, although it improved function in the MND ALS mice similar to dichloroacetate, it wasn't actually able to improve survival. And this outcome, I reasoned, was because we probably shouldn't be inhibiting the way that cells can access fat altogether, because they must be switching towards fat as the energy substrate for a reason. So if we inhibit their ability to use the fuel that they're wanting to use, this might have detrimental consequences. And so in 2019, we decided to try trimetazidine. Trimetazidine, similar in some way to ranolazine, is approved by the EMA for the treatment of angina. And again, it works at a very similar point of the mitochondria as ranolazine, but it is only a partial inhibitor of fatty acid use. So unlike ranolazine, which completely shuts down the cell's access to fatty acids, trimetazidine only partially shuts it down. So the cell can access both sugar and fat, but the idea is that trimetazidine might just tweak the way that the cells access both these fuel substrates. And so, as I said, we wanted to know whether metabolic flexibility was a therapeutic target. And this is why we came up with trimetazidine. Because it's EMA approved, it's used to treat angina, so we know that it's fairly safe and tolerable in humans, and that it is able to improve the way the cells access sugar. And so the first thing really was to see whether or not it did anything in the SOD mice. And what we have here are 4 graphs. The top are the wild-type non-MND mice, and the bottom are the G93A MND mice. We've got mice that are treated with trimetazidine, which are the light pink for the wild types, and the mustard lines for the ALS mice. On the left are the male mice, and on the right are the female mice. And what I hope you can appreciate from this graph is that the mice that are receiving trimetazidine, at least the ALS mice, they have lower oxygen consumption than the ALS mice that aren't receiving trimetazidine. So what this graph really shows is that trimetazidine is reducing this hypermetabolism that we see in the SOD1 G93A mice. What was also really important is that we looked at the motor neurons in the spinal cord of these mice. So we took spinal cords at 120 days, which is when the SOD1 mice have quite a significant MND phenotype, and 150 days of age, which is when mice are usually euthanized due to welfare reasons and the disease is quite progressed. But regardless of which stage we took the spinal cords, we noticed that the SOD G93A mice that were on trimetazidine had more motor neurons in the spinal cord compared to the G93A mice that did not receive trimetazidine. So not only did trimetazidine reverse or prevent hypermetabolism, it also prevented the death of motor neurons. But most importantly, what we showed was that trimetazidine was able to improve survival in the SOD1 mice. On the left, again, we have male mice, and on the right, we have female mice. The ALS mice receiving trimetazidine are denoted by the mustard lines again, and those ALS mice not receiving trimetazidine are the blue lines. And you can see that there's this rightward shift in the survival curve, meaning that the treated mice are living longer than the untreated mice. Now, trimetazidine seems to be doing amazing things in mice, but does it do anything in human cells? So this is work that's unpublished at the moment, but was generated by Leanne in the lab, where she took those stem cells that we generate from the skin samples from people with ALS and without ALS, and we turned them into motor neurons. So these are the motor neurons. They're stained. The nucleus is blue and the marker for the motor neuron is green. We can see that these are the no treatment cells, and these are the neurons that have been treated with trimetazidine. And we show that trimetazidine at lower doses to higher doses can actually improve the survival of these stem cell-derived motor neurons in culture. Now, in parallel to Leanne running these IPS studies, we applied for funding through FightMND to run a clinical trial to look at the safety and tolerability of trimetazidine in people with ALS. And this was based off of the preclinical data that we generated in the mice. Initially, this trial was slated to be run over Australia, the Netherlands, and the UK, but with COVID and Brexit, we ended up dropping the UK from the study, and we went ahead with Australia and the Netherlands. So the recruitment of participants into this study was led by Rob Henderson at the Royal Brisbane and Women's Hospital and Leonard Vandenberg at the University Medical Centre Utrecht in the Netherlands. We completed this study in July 2023. So this is what the METFLEX study looked like. We had people coming in for a screening visit at week 0, and those who met the eligibility criteria for the trial came in for a baseline visit 4 weeks after the screening visit. At the baseline visit, they received the trimetazidine, which they took twice daily, and they took that drug for 12 weeks. During that 12-week on-treatment period, we had 4 follow-up visits, with 2 of those being by phone and 2 in person. After the 12-month treatment period, there was a washout period of 4 weeks, and then we did another clinical visit face-to-face. Now, everyone who was enrolled in the trial received trimetazidine. So this was open label. There was no placebo control. So we screened 29 people for eligibility, and we excluded 8. 7 of these individuals didn't meet the criteria. 1 declined to participate. We eventually enrolled 21 into the study, and 2 people discontinued treatment, 1 due to disease progression, and 1 withdrew consent before starting the study. 19 of the 21 individuals completed the on-treatment period, but 4 discontinued the follow-up, and this was mainly due to disease progression and, with COVID, sometimes inability to come into the clinic due to restrictions associated with COVID. So overall, 15 of the 21 who were enrolled completed the whole study, but we analysed data from all 21 individuals who were enrolled, accounting for the dropouts. Overall, trimetazidine was very, very safe and very tolerable in people with MND ALS, and this really wasn't unexpected because of what we'd known previously and the fact that it is approved for use for angina in Europe. There was one individual who presented with some stiffness that may have been associated with the treatment, but they were self-limiting. So when the person went off the treatment, that stiffness was resolved. So overall, trimetazidine was very safe and tolerable. We wanted to look at biomarkers in the blood that might give us some indication of the fact that trimetazidine was acting as it should have been. And because trimetazidine's mechanism of action is to reduce the amount of fat that a cell is accessing for energy, we wanted to measure byproducts of fat oxidation or fat metabolism. And the 2 that we picked are MDA, malondialdehyde, and 8-hydroxydeoxyguanosine, or 8-OHDG. What we've got here are the graphs showing that we've got the screening period, and then 0 to 12 weeks is the on-treatment period, and then 12 to 16 weeks is that washout period. And what we showed is that while on the drug, measures of MDA decreased slightly, but not significantly in people on the trial, whereas 8-OHDD decreased significantly during the on-treatment period. And during the washout, the levels of 8-OHDG in the blood actually rebounded back to baseline levels prior to the treatment period. And what was really exciting for us was we also looked at energy expenditure in these individuals in the trial. And again, we've got the lead-in period of -4 to 0, the on-treatment period of 0 to 12 weeks, and then that washout period from 12 to 16 weeks. And what we saw was that the energy expenditure, or that resting energy metabolism in people with ALS on trometazidine, decreased significantly while they were on trometazidine, and it rebounded back up during the washout period. And this metabolic index is the term that we use to measure hypermetabolism. And we showed that hypermetabolism also decreased. So we're able to recover or prevent or reverse this hypermetabolism in people with ALS, MND with trometazidine. And again, this sort of rebounded back up close to the starting levels after the washout of the drug. And so where we hope to go next is to run METFLEX 2. So we know that the drug is safe and tolerable, and we know that it seems to decrease markers of fat metabolism, but also hypermetabolism in people with MND ALS. And so the next trial that we really want to run is to see whether or not trometazidine is improving clinical features of the disease in people across the world. We want to show that it improves ALS, FRSR, neurofilament, and potentially survival. And so we hope to run a multi-center study across Australia, mainly East Coast, unfortunately. Hopefully we can get the West Coast and Tassie on board. We've also got Leonard again in the Netherlands, 2 sites in the UK who are interested at Sheffield and King's College, 3 sites in Italy who are interested, and we've got some interest from Spain and Belgium at the moment. So hopefully we can start this trial at some stage with funding to support. So with that, I'd like to thank everyone for their attention today, and hopefully you found this insightful and informative, and I'm happy to take any questions.
Thanks so much, Shu. That was a really great talk, and really great to hear about this journey from a basic lab discovery through to hopefully a phase 2 clinical trial soon. So if anyone has any questions, I can start off. We've got Lizanne Uy. I'll just allow your mic and camera, Lizanne. Yep, that should be enabled now. So step up.
Yep, I'm here. Thanks.
Hi, Lizanne.
Hey, thanks so much, Shu. I mean, it's just beautiful work to see, the hypothesis being borne out all the way through and up to clinical trials. And so, fantastic to see that. So I think it's always really interesting when you're able to see a phenotype in the SOD1 mouse model, but also in human patient-derived cells. And so my question is, in thinking about, the next stage of your trial, do you think that it will be efficacious in all patients, or do you think that you need to stratify based on some kind of genetic markers or some kind of phenotype that you could measure first?
Yeah, it's a great question and something we've thought about quite a lot. In our 2018 study, even though it was small, we did show that the hypermetabolism was not really linked to any gene mutation, although admittedly it was a small study sample size. And so we're currently collaborating with Amar Al-Shalabi to look at the potential genetic underpinnings of metabolic phenotypes in MND ALS and whether the genetics can give us some information to help stratify. It's hard to say at the moment whether trometazidine would be beneficial for everybody or a specific subtype, but we hope that through a bigger phase 2B trial looking at the efficacy over many more people, that we might be able to tease some of that information out.
I mean, is there an opportunity or a possibility to test hypermetabolism first and then see if they might be a good candidate?
Yeah, so we did do that for the first trial where hypermetabolism was one of the inclusion criteria. But given that I guess, the incidence prevalence of hypermetabolism in cohorts can range anywhere from 30 to 60%, it really does limit recruitment into the study. And in some ways, you know, it's a bit of an exploratory biomarker where we really want to know whether the drug itself is helping people, not just metabolism. I think we would be remiss to exclude people because they're not hypermetabolic, although part of the trial design would involve analysis of the people who do have that hypermetabolic phenotype to see if they might have better outcomes overall compared to everyone else who may not have that hypermetabolism.
Yeah, great. Thank you.
Thanks, Lizanne. Any other questions from the floor? Sina, I will allow your mic and camera. There you go. Sina, please ask your question. Yeah, Sina, you should be able to unmute yourself now.
I'm saying Sina doesn't want to talk to me.
Well, I might ask a quick question just while we're trying to sort that out. I just wanted to ask, you've made a lot of wonderful collaborations around the world with some real leaders in the field from early on in your research career. So just a more general question, especially for some of our younger audience members, like how you approach leaders like that and start to build those sort of collaborations to really enhance your research.
Yeah, it was really that three very different stories actually. With the Jean-Philippe Lafleur, he was really a pioneer in this area in the preclinical space, and I just went to a conference overseas in Amsterdam at the time and invited myself to his lab. So he didn't really have a choice, and so I just had to be bold and take the opportunity. The worst he could have said was no, but he had me visit, and that's been a very successful and I think fruitful collaboration over the years.
Yeah.
With Leonard, I met him at a conference in 2013 in Milan, the international conference for ALS, and I actually didn't know who he was at the time because I wasn't really I'm not a neurologist, so I didn't know all of the neurologists around the traps, but it just turned out that he was coming to Australia over Christmas, so I offered to pick him up at the airport, and and it went from there. We just after picking up him from the airport and spending a day or two with him, I Googled him and realized who he was and thought, oh my gosh, did I say stupid things? Does he think I'm weird? But that also worked out okay, and we submitted a grant together. And then with Amar, it was actually my mentor, Naomi Ray, who introduced me to Amar. And he came over in 2016 and we took him through our natural history study. And he was really keen to collaborate because he really believed that looking at metabolic phenotypes was something of promise in the disease. So, yeah.
I guess the take-home message is don't be too afraid. They're not going to bite.
They're not going to bite, and sometimes you have to make your own luck, I think. Yeah, they're all really great people.
Sina, how are we going? Perhaps if you're having trouble, you can just type your question into the chat as well. Sina, I can read it out for you. And speaking of which, we do have a question in the chat from Brenton. It says, hi Shu, great talk. Can you comment on why fatty acid metabolism is ramped up in muscle? Are there defects in glucose transport or insulin levels normal?
So Brenton, great question, and we've asked that ourselves many times. It's in terms of glucose transport and insulin, I would say sensitivity. It's hard to say. There have been studies that have sort of reported both ways, that yes, there's decreased glucose transport and changes in insulin sensitivity, whereas other people have argued the opposite. So I think in the mouse model, it's really hard to know. In people living with MND, we've noticed some very interesting findings. I think with some initial glucose responses in patients that have been part of the natural history study, which is something we're trying to disentangle. But it's not, I don't think it's the cells can't use glucose, because when we force them back towards using glucose, they do it just fine. So it's just trying to figure out what is that switch and why is there the switch. There's a PhD student in the lab trying to work on that at the moment. So I'd say that we still really don't know. And it's something that we're trying to figure out through our preclinical work.
Thanks. Can Joe Nicolazzo, you should be able to unmute yourself.
Hi, thanks, Sheila. That was a really nice presentation. I've got two questions. The first one actually is similar to the last question. Have you looked at the fatty acid transporters in either the muscle cells, in the muscle cells, to see whether they're dysfunctional and whether the treatment affects transporter expression, function, mitochondrial transporters as well?
Yeah, so we've had a PhD student, Lauren, working on that for some time now, not just in the context of just fuel use, but also in the context of fatigue and how the fatty acid transporters might change. Now, classically, when looking at the transporters, people have done just, protein expression or gene expression. We've been trying to look more closely at the function of the rate-limiting enzymes in the glucose and fatty acid pathways, which is challenging in itself because of the amount of fresh mitochondria that you need to isolate to be able to do it. So, we're just trying to figure out the most efficient way to answer these questions by looking at the enzyme activities.
Okay.
Yeah.
Okay. Nice. And the second question is, I know you've been focusing on the muscle cells, and you showed that if I got it correctly, that trimetazidine didn't have any effect on the neurons. But other cell types, involvement of other cell types in the beneficial effects of trimetazidine, microglia, astrocytes.
Yeah. So
and friends.
So we'd shown that trimetazidine improves motor neuron survival in the mice, but also our iPS-derived motor neurons. And there's been a recent paper by my colleagues in Italy showing that the trimetazidine does work on the spinal cord, and it reduces the activated microglia in the spinal cord. So I think it's not just an effect on muscle and neurons. I think any cell that requires glucose fatty acid flux for energy substrates to fuel any function would be impacted by trimetazidine, and it's to what degree these cells respond potentially to trimetazidine throughout the course of disease, because because the disease changes, although very defined in the mouse, I think in people with MND, it's going to be something that needs to be tracked quite closely, because what might work at one time may not work further on in the disease based on what's happening at the cellular level in the spinal cord and brain.
Yeah. Thanks very much.
Thanks, Joe. I have seen his question typed into the chat here, Shu. He says, "Hi, Shu, fantastic talk. Have you considered the effects of a ketogenic diet and ketone bodies?"
I'm not a dietitian, so I won't comment on ketogenic diets, but we do have a dietitian working in the team who I think would suggest, and I think Derek would suggest too, that ketogenic diets are probably not advisable in MND ALS, because they're really geared towards weight loss and fat loss, which is something that leads towards prognosis in MND ALS. So I don't, and the studies that have been done on ketogenic diets thus far have really been preliminary and not done well, at least in patient populations. So I think at the moment, the recommendation would be no to the ketogenic diets and ketone bodies.
Thanks. Janet? So could you just unmute yourself as well, Janet? There should be a little mic icon in the top right. I'm sorry, I can't do much. Again, I've allowed your mic, so there's not much I'm able to do. Would you like to type your question into the chat instead, Janet?
But Sina, as a follow-up to that, we did try medium chain triglycerides in SOD1 mice, and that did improve function and survival to some extent. There was a phase 1 trial run on these medium chain triglycerides, triheptanoid in particular. That was done by Richard Bedlack. But the high fat content of that diet was also made it not very well tolerated in people with MND, given that there are some gut issues reported with people with MND already.
Okay. So I have a question from Sunny Ann in the chat. It says, "Hi Shu, it was a great talk. Similar to Sina's question, the SOD1 mice, were they fed with a chow diet? And have you checked fat versus lean mass with total mass from these mice?"
So in terms of high-fat diets, they have been used in SOD1 mice, and high-fat diets do improve survival and outcomes in the SOD1 mice. In terms of fat and lean mass, well, as mice get to about the onset of disease, from onset through to mid-stage, they lose a lot of lean mass. We've done that through NMR. And they also lose fat mass. So they're gaining fat mass, and then they get to a point where no matter what they're eating, they're failing to put on extra weight. And that is a combination of not being able to put on extra fat mass and potentially losing fat mass because the muscle is turning to that stored fat as the energy substrate, but also a concurrent loss of the lean mass, the muscle mass. So it's a loss of all masses altogether.
Thanks. Janet, did you want to give it another go or type your question? I think if there are no more questions from the floor, we might wrap it up. Oh, sorry, one more question. Sorry, Shu. Another question from Brenton Short. Would insulin injections help to get more glucose into the muscle and abrogate the fat metabolism if used with the fatty acid inhibitors?
Brenton, I think it may do up to a certain degree, but I think there might be some insulin insensitivity in these animals due to the change in mitochondrial function. What we have done, and a paper that's coming up in the pipelines for us, is we did cross the SOD1 mouse to a mouse that is very, very highly insulin sensitive. So its glucose metabolism is just absolutely pristine. And what we've noticed in those mice is that it delayed the onset of the disease by, if I recall from memory, 8 to 10 weeks. But the problem was that once the mice developed the disease, they weren't able to mobilize the fat stores, and they died at the same age as the other mice. So although it delayed disease onset, the progression of the disease was very, very rapid. And so this really shows us that we need to tweak the access to glucose and sugar throughout the course of disease.
Thanks, Shu. I think we might wrap it up there. I'm sorry, Janet, that your mic didn't respond there, but please, I encourage you to email the question through to us at FightMND, and I'm very happy to pass it on to Shu, who I'm sure will be happy to answer it. And if anyone has anything else they'd like to follow up, please feel free to reach out to us at FightMND, and we can connect you through to Shu, or she'd be happy to receive an email from you separately. So I'd really like to thank you for your presentation today, Shu. I think it was really particularly interesting to show that there's a pathway to turn your early discoveries into really tangible outcomes like a clinical trial. So yeah, thank you for joining us today, everybody. The April FightMND scientific seminar. The next monthly seminar will be on Tuesday, the 27th of May at 12 p.m. Australian Eastern Standard Time, and we'll release the details in the next couple of weeks. So please keep an eye out in your inbox for this. Until then, all the best, and we'll see you next time.
Thank you, Sarah. Thank you, everyone. Have a good
you.
afternoon.